students, coming to the next class. We already known about this topic, that's why I'm getting laugh, right? See, structure of chloroplast. Now first year we discussed about the chloroplast structure. Based on that, within that chloroplast there is an outer membrane is there, there is an inner membrane is there, right? See, outer membrane and inner membrane, between these two there is an periplastidial space is present, right? See, that is the one of the space, right? After that, coming to the inside the chloroplast structure, there is a plate-like structures are there, right? See, these are the individual plate-like structure. A row of plate-like structures are called as granum, right? See, this is one of the thylakoid membrane, and this row is called as granum. Total, okay, this is a granum, this is a granum, this is a granum. The total content is collectively called as grana. The total content is called as grana. Remember what I am telling here, there is a singular is there, plural is there, right? Here the singular indicates here granum, plural indicates here grana, plural indicates here grana, right? So that is the clear statement regarding to that granum and the grana. So row of thylakoid membranes are there, row of thylakoid membranes which are called as granum, granums collectively called as grana, right? See, these are the sites for the photosynthesis process. These are the sites for the photosynthesis process. So, which are involved in the light energy trapping process. The thylakoid membranes which are involved in the light energy trapping process. Simple. See, the light energy is converted into that chemical energy done by that thylakoid membrane, because within that thylakoid membrane, we already discussed there is an PS1 means photosystem 1 and PS2 photosystem 2 is there. Right, these are responsible to trap that light energy. Again, within that systems, along with these systems, some of that other components also there, right, which are also supporting for the trapping of light energy. Finally, see, the light energy is converted into chemical energy. So here, this is a chloroplast structure. Like that, I'm expecting I need not to write all these parts, but outer membrane, this part is called as outer membrane, right? And this is the inner membrane, right? This is inner membrane, right? So individual plate-like structure is there. This one is called as thylakoid. This is the thylakoid membrane, right? The individual structure is called as thylakoid membrane. Right here there is an row is there. The row is called as granum. The row is called as granum. The total granums are called as, means the total content is called as, see, this is a singular, this is a singular. The total content here called as grana. This one is indicates plural. This one is the singular, this one is the plural, right? So that is the big difference between granum and the grana, right? So here, see here within the chloroplast structure, we have to discuss about one of the important site is the stroma. Okay, stroma part is also very important. See, whatever that other than that thylakoid membranes, right? See, whatever that fluid content is present inside the chloroplast, the content is called as stroma. Then we have to divide it into the photosynthesis process. There is in two phases. We already know. Okay, that one is called as light reaction. Other one is called as dark reaction. Light reaction takes place within the thylakoid membrane. Dark reaction occurs within the stroma. So this is a big difference between the thylakoid membranes. Light reaction and the dark reaction. So here concept is thylakoid membrane is responsible for the trapping of light energy. Then the stroma is responsible for the dark reactions. Means, see, the carbon dioxide reaction done by the stroma, or the carbon dioxide reaction done by the dark reaction. So here the concept is there is in granum is there, another granum is there, right? Between these two granums, some of the connecting structures are there, which are called as stroma lamella. Okay, these extended parts, which are called as Stroma Lamella. Yeah, this part is called as Stroma Lamella. So Stroma Lamella also performs one kind of special function for the trapping of light energy, but the grana function is quite different from the Stroma Lamella function, because some of the additional functions are performed by the stroma, some of the functions are unable to performed by the Stroma Lamella. So what are the functions? Definitely we'll discuss. Right? See, this is about that Stroma Lamella, and this is about that grana or the thylakoid membranes, right? This is about that granum or the grana, right? So here the total structures are collectively called as grana, right? Okay. And all of you remember, within that stroma part, there is in ribosomes are there, and these ribosomes are 70S ribosomes. These ribosomes are 70S ribosomes, right? These 70S ribosomes are involved in the protein synthesis process. The 70S ribosomes are involved in the protein synthesis process. And coming to the next one, there is in blue color structures are there. These are nothing but starch granules. Right? These are called as starch granules. So whatever that glucose molecules are there, the glucose molecules are synthesized by the photosynthesis process. So within the plants, the storage form of carbohydrates is the starch. So within that chloroplast structure, there is in starch granules are there. So that one indicates within the chloroplast, the starch is stored, right? So these are nothing but starch granules. So this is about the chloroplast structure. The simple explanation: outer membrane is there, inner membrane is there, right? At the center part, there is in thylakoid membranes are there, which are plate-like structures. A row of plates called as granum, right? A group of granums are called as grana, right? See, the granum is connected with that another granum. That one, the connecting structure is called as stroma lamella. Stroma lamella function is quite different from the grana or thylakoid membrane part or the granum part, right? So this is this is about the big difference between these two. We'll discuss definitely. And see, there is in ribosomes are there, and storage granules are present, right? Not only these, some of the lipid droplets also present within the chloroplast. Some of the lipid droplets also present, and one more important thing is the chloroplast having that own DNA. I didn't draw here because all these parameters we discussed within that first year. That's why, right? This is one of the this is one of that double-stranded DNA. That is nothing but plasmid DNA. So here double-stranded circular DNA is present within that chloroplast structure, right? So that one is called as Eukaryotic Plasmid. So I already said plasmids not only present in the prokaryotic cells, the plasmids also seen in eukaryotic cells too, right? So within that eukaryotic cells, the mitochondria and the chloroplast also having that plasmid structures. So the plasmids and the 70S ribosomes are evidences for the evolution of mitochondria and chloroplast from the prokaryotic cells, right? So within the chloroplast, the functional performance is there is a light reaction is there, dark reaction is there. We are already well known about this one. The light energy is converted into chemical energy, right? See, end of the light reaction, within the chloroplast structure, there is an ATP production takes place, and NADPH2 production takes place. Remember, I am talking about end of the light reaction. End of the light reaction, there is an ATP production takes place, and NADPH2 production takes place. Right? Remember, all of you, there is an oxygen evolving process also occurs within the light phase, within the light reaction. The oxygen production process also occur. Then we know about that oxygen evolution process, and remember in the last class I said photolysis of water. There is an Hill's reaction we discussed, right? Splitting of water molecules, that is nothing but the Hill's reaction. So the Hill's reaction is also done within the light phase or light reaction. Within the light reactions, photolysis of water takes place. Oxygen evolved, right, within that light phase, right? Next one is that ADP synthesis also done within the end of the light phase, right? And next one is the NADPH2 production also takes place within the light phase, right? So that is about some of the products of the light phase. Remember, whatever that dark reaction is there, the dark reaction not depends on light, definitely, because there is no light requirement. There is no light requirement for the dark reaction. Either presence of light, either absence of light, okay, the dark reaction will be takes place. But light reactions are not like that. Light reactions always occurs presence of light only, right? So here there is in components which are present, grana and the stroma I mentioned here. So within the end of the dark reaction, within the end of the dark reaction we get the glucose, right? So we know about that the glucose reduction process within the dicot plants. There is an C3 cycle is there, right? And within the monocot plants there is an C4 cycle is there, and within the xerophytic plants there is an CAM cycle is there, right? So these are the different cycles present in different plants, because the plants are adapted, right? Based on that adaptation process, these cycles are occurred. So definitely within the dicots, the C3 cycle is the C3 cycle involved in the glucose production, right? C4 cycle seen in within the monocots, right? And the CAM cycle is there. The CAM cycle is seen in the xerophytic plants, right? So based on adaptations, the cycles are different. So why one is C3, why other one is that C4? See, C3 means the first compound within the C3 cycle, okay, contains that 3 carbon molecules. So that's the reason why that one is called as C3 cycle, and other one is called as the C4 cycle, because the other one, especially the first stable compound or first compound, okay, within that cycle is the C4 compound. That compound having the 4 carbon molecules, right? So based on that, we can say there is in C3 cycle and there is in C4 cycle is there. C3 cycle is seen in dicot plants and C4 cycle is seen in the monocot plants. Crystallization acid metabolism, because CAM cycle, okay, especially this cycle is seen in xerophytic plants. So within the dark reactions, maybe the C3 cycle, maybe the C4 cycle, and maybe the CAM cycle. These are responsible for the reduction, right? So, and I like to discuss about the components which are present in the grana and the stroma lamella and which are responsible functions, right? See, the PS1 photosystem, photosystem 2 first I mentioned here, PS2 I mentioned here, right? So PS2 present within the grana and the PS2 absent within the stroma lamella, means this part is called as stroma lamella, we know this part is called as stroma lamella. So these structures, these are nothing but the total contents are called as grana, right? So thylakoid membrane or the granum, right? We can name anyone here, either maybe the grana or granum or the thylakoid membrane, right? So within these structures there is an PS2 is present, but within the stroma lamella, right, the PS2 is absent. So PS2 is the very important component for some specific functions we'll discuss. See, PS1 is there, PS1 is present within the grana and within the stroma lamella, right? So, and electron transport chain, that one is also seen within that grana and the stroma lamella, but the type of electron transport chains are different. See, within that grana there is a jet scheme, okay, mechanism is there. Jet scheme mechanism is nothing but electrons are transported in the jet, okay, the total structure resembles the jet shape, right? That's the reason why, see, the electron transport chain is called as jet scheme mechanism. And within the stroma lamella, right, there is in cyclic, the transportation is called as cyclic. So within the stroma lamella, the electrons are transported in cyclic manner. The electrons are transported, okay, in cyclic manner, right? ATP synthesis process. So here we know mitochondria involved in the ADP synthesis process. Chloroplast also involved in the ADP synthesis process. Remember, mitochondria produce the ATP, utilize it for the cell functions. Whatever that cell needs are there, cell requirements are there, all the functions, okay, done by the mitochondria produced ATP. But whatever the chloroplast is there, the chloroplast produced ATP is there, that ATP, remember, okay, only utilized for the photosynthesis process only. This ATP is only utilized for the photosynthesis only. So here grana also involved in the ATP synthesis, Stroma Lamella also involved in the ATP synthesis, right? That is nothing but jet stream mechanism also involved in the ATP synthesis. Cyclic electron transport is also involved in the ATP synthesis, right? So coming to the next one is that NADPH2 production. Remember, okay, in the catabolism process, NADH plus H plus is produced. In the anabolism process, NADPH2 is involved. NADPH2 is involved. So here NADPH2, okay, is produced, and that one is the NADPH2 is involved in the synthesis of glucose molecules. That is nothing but anabolism. So here NADPH2 is produced, right? See, the NADPH2 production done by the grana only. Stroma lamella is unable to produce the NADPH2, right? And oxygen splitting process is only done by the grana, right? See, oxygen splitting process, we know that Hill's reaction, that is nothing but the photolysis of water done by the only grana, right? See, the Stroma lamella unable to perform that photolysis of water. Oxygen evolution complex means oxygen production, right? See, this is nothing but oxygen evolution complex and oxygen evolution. If complex is present, oxygen is evolved. If complex is absent, okay, oxygen is not evolved. So here, grana have the capacity to produce the oxygen because it's having the oxygen evolution complex. So within the grana, oxygen evolution complex is present and it evolves the oxygen. Within the Stroma lamella, okay, oxygen evolution complex is absent. It's unable to produce the oxygen because oxygen evolution complex is absent. So if we carefully observe this pattern, okay, the total contents, see the total, okay, water splitting process, oxygen evolution, right, NADPH2, these three are linked with two, okay, these three are particularly linked with two, okay, one component, see, NADPH2, splitting of water, right, oxygen evolution, complex, and produce the oxygen. These three are linked with the particular one component. See, the component is, if we observe carefully, the component is the PS2. If PS2 is present, okay, NADPH2 production takes place. If PS2 is present, splitting of water takes place. If pH2 is present, okay, oxygen evolution takes place. So here, what I am telling, the PS2 is responsible. For some specific functions. That's the reason why when I am explaining regarding to that PS1 and PS2, right, and when I am explaining regarding to that grana and the Stroma lamella, some quite differences are there for the functional performance. Okay, these are the major differences. See, NADPH2 synthesis done by the PS2, presence of PS2, and splitting of water done by the presence of PS2, right? See, oxygen evolution complex presence of PS2. So if PS2 is present, okay, majority of the functions are performed. If PS2 is absent, majority of the functions are unable to perform. So within the photosynthesis process, PS2 is very important component to perform very perfect or very important functions. Right? So that is about the differences between, functional differences between the grana and the Stroma lamella. Right? Coming to the next one, we will discuss about the next topic. Right? Coming to the next topic is isolation of plant pigments from the leaf extract. See, we are isolating the pigments. So how we are isolating plant pigments from the leaf extract? See, we are using one of the technique. The technique is called as paper chromatography technique. All of you remember we know, we know the leaf structures, and the leaf exhibit green color because of the chlorophyll. Okay, basically some leaves exhibit different colors because dominated pigment, based on the dominated pigment, the color will be changed. So here also there is in leaf pigments we try to isolate. For that purpose we have collected some of the leaves, right? So we are subjecting for the pistol and mortar. Pistol and mortar, that is nothing but one of that bowl is there and one of the mortar is there. We collected some of the leaves, right? The leaves are added to that bowl structure. We are applying mechanical pressure and we are adding 90% of the acetone. We added one of the liquid, 90% of the acetone, and we added some of the leaves, right? And we started that grinding process. After completion of grinding process, we get the sap. We get the sap. The sap is nothing but there is a green color liquid. Finally, we get after complete grinding process, green color liquid, right? That one is called as pigment sap. That one is called as pigment sap, right? That pigment sap we are separating for the, we are subjecting for the separation process. We are subjecting for the separation process. That is nothing but paper chromatography technique. The technique is called as paper chromatography technique. For suppose one of the paper, if we collect, just we taken one paper, right? But in the paper chromatography, normal papers are we are not using. Wattman number 1 filter paper we are using. That is the pure cellulose content paper. That's the reason why we are using that Wattman number 1 filter paper. So once if we collect that Wattman number 1 filter paper, whatever that sap we collected, one drop is taken. Just imagine this is the paper. Okay. So the bottom side of the paper we spot the sap solution. So this is one of the paper, right? We collected. Okay, already we prepared the sap solution. One of the drop is taken and we spot at the bottom of the paper. We spot at the bottom of the paper. After the drying process, after the spotting, we need to wait up. After the drying, right? Once the spot is dried, then the paper inserted inside the chamber. Inside the chamber, right? See, the chamber contains one of the liquid-like solution, not like one of the liquid solution. The chamber contains one of the liquid solution, right? So this is one of the liquid solution. We will insert the paper inside the chamber and inside the liquid, right? Once, if we kept the paper inside the liquid solution, then the pigments starts the moving. So the pigments are moved from spotted place to opposite direction. Opposite direction means this is the spotted place. So the pigments are moving. From here to here. Because here the liquid is there. The liquid is act like a mobile phase. So liquid is supporting for the movement of plant pigment. See, the liquid is supporting for the movement of plant pigments, right? See, whatever the paper is there, the paper is called as stationary phase. So we are separating molecules in between two phases. One of the phase is called as mobile phase, other phase is called as stationary phase. So based on these two phases, whatever that pigments we are subjected, means here plant pigments we are subjected, we are separating the plant pigments. So see, there is an orange color band we get after the separation process, yellow color band we get, right? Green color band we get, and blue-green color band we get. So normal green is there and blue-green is also there, we get. So here we get that 4 types of the bands. Definitely we have this experiment in our practical also. We need to do this experiment. This is one of the experiment for the final, for the practical examination point of view. So within that mobile phase, so we are using here mobile phase. So how we prepare the mobile phase? Here there is a 90 ml of petroleum ether, 90 ml of petroleum ether, right? And 1 ml of acetone, 1 ml of acetone. So that is the mixture. So here for the mobile phase we are using a mixture of solution, that is nothing but petroleum ether and acetone. One is that 9 ml, other one is the 1 ml, right? So that is the one of the mixture of the solution. That one we used as mobile phase. That mobile phase is supporting for the separation of the pigments. So here there is an orange color band, yellow color band, green and blue green. Sir, why here the colors are different? Okay, we'll discuss. Stationary phase. Stationary phase is nothing but paper. Sir, how paper is act like in stationary phase? Remember, the papers are prepared by the cellulose. Already this is the Wattman number 1 paper. Wattman number 1 paper. This is the pure cellulose contained paper. So here cellulose molecules are act like in stationary phase. Cellulose molecules are act like in stationary phase. So after that separation process, after drying the paper, we get these colors. In that case, the very top color is the very top. Color is the orange color. See, orange color indicates here carotenoids. See, the molecular formula for the carotenoids is the C40, right, and H56. C40 and H56, right? And below that orange color, we get the yellow color. We know that yellow color indicates here there is a xanthophylls are there. That xanthophylls, having the molecular formula is C40, H56 and O2. See, additionally here oxygen molecules are present. Here oxygen molecules are absent. That's the difference, right? And next one is there is a chlorophyll A is there. Within that, the chlorophyll A, the color is here green color. See, the third band is the green color. The green color band indicates here chlorophyll A. Here molecular formula. Remember, magnesium is the metal ion. No doubt within the chlorophyll structure. There is a C55, H72, O5, N4 and magnesium ions. Remember, there is a nitrogen atoms are there, oxygen atoms are there, hydrogen atoms are there, carbon atoms are there, and magnesium is the central ion. Central metal ion, right? So magnesium also present within that chlorophyll A structure. And coming to the next, that is the last band, that is the bottom band. The band color is the blue-green color. Here there is, that one is nothing but chlorophyll B. Here components are C55, H70, O6, N4, Mg. Remember, if we observe the differences between chlorophyll A and chlorophyll B, here H72, here H70, here O5, here O6. Remaining all are same. Only difference between hydrogen atoms and the oxygen atoms. That is the only difference. Remaining all are same. So that is about that 4 colors of the bands, and we got here 4 colors of the pigments. That is about that separation of plant pigments by the paper chromatography technique. Within the technique we are using Wattman number 1 filter paper. Remember, here 2 important phases are there. One of the phase is called as mobile phase. For the preparation of mobile phase we are using 9 ml of petroleum ether and 1 ml of acetone. And other phase is called as stationary phase, right? The stationary phase is supported for the separation of the plant pigments because cellulose, right? All of you remember, after that spotting, after the spotting of the sap extract, right? After the pigment sap or sap extract, then the whatever the pigments are there, the pigments are separated. The pigments are separated. So this technique is called as paper chromatography technique, right? That is about the isolation of plant pigments from the leaf, right? By the extraction method. Remember, simple, we are using one of the acetone. After the grinding process we get the one of the pigment sap, right? That is subjecting for the separation. That is about the separation of plant pigments. Separation of plant pigments, right? Next one, we will discuss about the next topic.